So my first task this morning is going to be to persuade you that a talk about evolutionary biology in Arabidopsis might actually be relevant or at least interesting to most of you in the audience. And I'll try to do that by just offering the classic definition of local adaptation, which is the focus of today's talk. So classically, local adaptation is thought of being a situation in which local genotypes have a higher relative fitness in their home environment than do foreign genotypes in that environment. And a lot of evolutionary theory tells us that the genetic architecture of adaptive traits can have a strong influence on the efficacy of selection in future modification of those traits. Now, by analogy, here, let me get rid of this thing. Sorry. By analogy, we might argue that local land races or cultivars or elite varieties or whatever will have a higher average yield than land races or cultivars originating in other environments. And then that the genetic architecture of traits of interest in an agronomic setting can strongly influence the efficacy of artificial selection in future modification of those traits. So I think one lesson we've learned from model systems like Arabidopsis so far is that it may not necessarily be fruitful to take our lists of genes we think matter in Arabidopsis and try to translate those to crop systems. But my hope, at least, is that some of the principles about how those genes are controlled sort of in a general sense might be able to be more translatable to crop systems as we try to design optimal breeding strategies. Arabidopsis, of course, is one of our best plant model systems. And some of the things we benefit from an evolutionary perspective of understanding evolutionary processes is that for many years people have been collecting Arabidopsis from all over the world, both in its native and its introduced range. So we now have thousands of collections available that have been inbred and we can be distributed to the research public. And along with that, of course, we have abundant genomic resources. So one thing that was quickly observed when we brought back Arabidopsis from these diverse locales and grew them in the common gardens of our growth chambers was that there's actually a tremendous amount of morphological variation among those lines. What's less well understood is how much physiological variation is among those lines. And particularly, what we're missing is an understanding of whether or not these differences are adaptive, which is to say, do the phenotypic and physiological differences we see between accessions actually have an adaptive value at a local level? Is there local adaptation in Arabidopsis? Most of the resources that have been collected have been used to dissect the genetic basis of traits without really thinking about the evolutionary context of them. Several recent studies, it's a little light, sorry. Several recent studies have begun to shed light on this issue, particularly this very nice study that came out just in the last 6 months, looking at a transplant between accession from the far north of the Arabidopsis natural range, native range, and the far south, so a reciprocal common garden experiment. Looking over 5 years, they were able to show that there is, in fact, this sort of classic pattern of local adaptation, which is to say that local environments, so here the Italian genotype and the Italian locale, show a higher average fitness than does the foreign genotype, in this case Sweden, and the reverse case is seen in the Sweden locale. What's particularly interesting about this study is to note that over the course of 5 years, the magnitude of that change, the magnitude of that difference is actually pretty variable. And just to draw attention to the last 2 years of the study, both in 2009, 2010, and the following year showed a significant genotype by site interaction and fitness. Well, in 1 year, and though in 1 year this was manifested in sort of the classic crossing reaction norm, where there's some sort of potential for an antagonistic relationship in the genotypes of the two sites. Sorry, there's evidence of trade-off between the two sites. In the second year, it looks as if at one of the sites there was no fitness difference between the two genotypes, where there was a large fitness difference in the second site. Taking a very different approach, 2 studies that came out back-to-back in Science last fall used a genome-wide association mapping approach to try to identify the genomic basis of local differentiation with respect to climate. Both these data sets made use of the same, both of these studies made use of the same large data set of about 215,000 SNPs that have been collected for over 1,000 Arabidopsis accessions. And the first study took the approach of looking for correlations between the SNPs and some climate variables they thought might have been relevant to local differentiation. And they then used those SNPs with significant climate associations to predict the fitnesses of about 150 accessions that they grew in a common environment in France. And they did, in fact, find that they were able to predict fitness with some reasonable certainty in that environment using their climate associations. Now, using sort of the reverse approach, the second group, the Fournier-Level paper, they grew about 150 accessions in 4 common gardens across the European distribution of Arabidopsis. They then measured fitness in those environments as estimated through fruit count and survivorship. And then using those fitness data, they found SNPs that were associated with differences in fitness. Now, needless to say, both of these studies found abundant SNPs that seemed to be associated with climate or fitness in the latter case. But where they differed significantly was in the role they attributed to selection in structuring that variation. The first group found considerable evidence for the action of positive selection or selective sweeps, which is, say, new variants are rising, being swept to high frequency due to the action of positive selection, whereas the latter group invoked more of a role for widespread genetic diversity that's largely where alleles are largely neutral in most locales, but then selected against in regions where they're deleterious. So two sort of contrasting ideas about the role of selection in maintaining adaptive differences within Arabidopsis. So as has been touched on several times yesterday and again, I'm sure today, one of the key things that we're all interested in is genotype environment interaction. And G by E is typically thought as being sort of a necessary prerequisite for local adaptation to evolve. What's probably less widely appreciated is that G by E can have several different patterns that can give rise to this interaction effect. And just to draw your attention back to this study I showed you a minute ago, we see on one hand these sort of classic crossing reaction norms, on the other hand this case of conditional neutrality. And this is true both at the whole organism level, in the case here with fitness, and also at individual loci. So individual loci can show so-called antagonistic pleiotropy or conditional neutrality. And finding which genes underlying which traits follow these two patterns, I think is an important goal for us as evolutionary biologists, potentially as it might influence how we think selection can act on these traits, both in the field and in a breeding program. So despite some recent work, is my water over there? Sorry. Despite the work I've just shown you, there are still quite a few unanswered questions, I think, in the genetic basis of adaptation. The first is we don't have a really strong sense, particularly in these widespread species such as Arabidopsis, how much fitness-associated traits are varying within a species. Nor do we have a really strong sense of which genes, and particularly which variants in which kinds of genes, underlie these phenotypic differences that are locally adapted. As I mentioned, there's still conflicting evidence for the role that natural selection plays in structuring local adaptation. And finally, I think we're just beginning to understand the role that phenotypic plasticity plays in local adaptation. So it might be intuitive to think that one highly plastic genotype might do well across all environments. And if that's the case, how does that influence the evolution of specialization in more difficult to adapt to, or more difficult to survive in habitats? So the work I'll show you today, I think, speaks a little bit to these first 2 points. And I hope we'll sort of begin thinking about these latter 2 questions. So what I'd like to share with you today is a 3-part story. First, taking a physiological genomic approach, combining physiology data with transcriptome data, trying to understand the drought stress response in Arabidopsis. We'll then take some of those transcriptome data and apply it to a genome-wide association mapping approach to try to understand the role of expression in structuring local adaptation to climate. And finally, I'll take a more targeted approach, looking at just one gene that we think is really important in the drought stress response in Arabidopsis, and trying to look at how natural selection has shaped variation in that gene. So my goal for the first part of this talk will be to try to identify whether or not natural accessions of Arabidopsis show variation in their physiological response to a mild soil water deficit. We'll then try to address what's the genomic basis of this variation. And finally, get a bit of a sense of what cellular processes are driving those responses to soil drying in Arabidopsis. So the goal here in this first bit is to combine transcriptome data with the physiological data to try to identify the genes and networks we think are driving phenotypic plasticity and possibly local adaptation. So to give you an idea of what the experiment entailed, we used 17 natural accessions of Arabidopsis from throughout the native range of Arabidopsis. 10 of these accessions were so-called spring annuals. These are genotypes that don't require a vernalization treatment in order to flower. And 7 were from so-called winter accessions, the late flowering accessions that require an extended vernalization period before they'll bolt and move to reproduction. I should note that we grew these two life history types in separate experiments. Just from a practical perspective, it was too difficult to harvest everything at once in a timely manner. Contrary to a lot of past experiments looking at Arabidopsis, drought, stress, transcriptomics, we grew these things in potting media, so hopefully a little bit more of a realistic environment than on, for example, MS plates. We subjected the plants to 2 treatments, a control, well-watered treatment, and a water-limited treatment, and the experiments were randomized, not nicely stratified by treatment as seen here. Now, I need to make it clear that all these measurements were made before bolting, so this all based on vegetative growth, and all the transcriptome data is collected from vegetative pre-bolting leaf tissue. So Jim Richards was one of the principal collaborators in this project, and he performed most of the physiological and developmental assays. So we looked at things such as specific leaf area, measured root-shoot biomass ratios. We used carbon isotope ratios as a measure of apparent water sufficiency. We looked at carbon-nitrogen ratios, and a number of leaf traits, including leaf osmotic potential, water potential, turgor, and leaf water content. I want to just give you a brief overview to convince you that we actually did impose a stress treatment here. So the top reaction norms are showing you the spring the spring experiment and the winter experiment under control and dry treatments, that we did in fact impose a fairly significant but a fairly mild drought stress treatment. So soil water potential sort of in the negative 1.2 millipascal range. This resulted in reductions in leaf water content in nearly all of the accessions. And of course, this resulted in reductions in osmotic potentials of the leaves. A couple of responses to highlight from the developmental aspect, looking first at root-weight ratio, contrasting the control environment in white, the dry treatment in gray. We can see that in general, there was a trend towards increase in root-weight ratio, and that's driven primarily by an increase in root biomass. The above-ground tissues were largely unchanged between the drought and the wet treatment, but the roots, the plants put on considerable more root tissue. Another interesting response we saw was in the amount of leaf nitrogen content. And this was one trait that in the spring annuals we saw a significant genotype by environment interaction, which is to say the different genotypes differed in how they accumulated nitrogen in their leaf tissue. And that was driven largely actually by Columbia, which is our wild type in Arabidopsis, which is one of 2 accessions to actually show a reduction in leaf nitrogen content in response to the drought stress. And another genotype, this line here, HR5, was the other oddball in that regard. Okay, so turning now towards the transcriptome data we collected in this experiment. Again, there were 17 genotypes across 2 treatments. We used 3 biological replicates in the transcriptome data. Our transcriptome estimates came from the Arabidopsis ATH1 microarray. It's important to note that we're dealing with very diverse genetic material here, and that we needed to exclude the possibility that just differences in the mRNA sequences were leading to differences in hybridization to the arrays. So as a side experiment, we hybridized genomic DNA to the arrays, found probes that were polymorphic, excluded those, and that leaves about a little over 20,000 probe sets, or about 20,000 genes represented in our transcriptome analysis. Just to give you a brief overview of the kinds of effects we saw in the transcriptome data. So we were able to identify 3 principal effects. First is so-called genotype effect. So these are transcription that just varies constitutively between the different accessions, regardless of treatment. We found about 8,600 genes that showed this constitutive genotypic effect on expression. We found about 6,700 genes that showed a significant environment effect. So these are genes that were either upregulated or downregulated in response to our drought treatment. We found about 1,000 genes that showed the interaction between genotype and environment. And this came in many different flavors. This came in genes showing crossing reaction norms, up in one treatment, down in the other, different between genotypes. And we also found a number of genes that showed this interesting pattern where there's very little difference in one environment, but a significant difference in the second environment. Another way of looking at this genetic diversity in transcription is just to look at the raw number of genes whose expression was affected by the treatment. So here I'm showing you genes that were either induced by drought treatment or repressed by drought treatment. And each bar represents one of the 17 accessions in the experiment, the spring annuals and the winter annuals. And I want to draw your attention to, first of all, Columbia here, which turns out is a pretty mellow responder in terms of just the number of genes that are affected by drought. So there's only about 1,600 genes that are affected by Columbia, and not a whole lot more in the other popular lab strain, Landsberg erecta. By contrast, some of the accessions, for example, this guy SQ8, showed over 4,000 genes responding to the stress treatment. So tremendous diversity just in how many genes are being turned up or down by stress treatment. Another way of looking at these data is to consider each individual transcript and see what the fold change was of each transcript. And again, there's quite a bit of variation. So for example, this TS1 genotype from Spain, no single transcript was up or down regulated more than 5-fold. By contrast, some genes, I think are lost on this figure, were over 20-fold induced or repressed in response to the stress. And again, this SQ8 guy sticks out as kind of an oddball. It has this huge collection of genes that were expressed more than 5-fold. So a really strong response across the genome to our treatment. In general, to summarize some of the functional observations from our analysis, we found a lot of genes involved in photosynthesis and primary metabolism, particularly starch synthesis and degradation, that were generally upregulated among our accessions, with a few noted exceptions. We generally find a paucity of things annotated as abiotic stress genes in Arabidopsis. It's important to note at this point, I think, that a lot of the genes that are annotated, particularly in TARE as abiotic stress, come from the Columbia accession from a small number of lab-based measures of stress response. And so it could be that those are not necessarily indicative of the sorts of soil drying that we're reporting here. We generally see an upregulation of these poorly understood pentatricopeptide repeat genes and downregulation of histone-related proteins. And there's many other interesting patterns in the data that I'm happy to share with you later. Another way of looking at functional enrichment is to assess whether or not particular predicted promoter binding motifs are overrepresented among the treatment-responsive genes. So these are genes that are responding to our environmental stress. And you can see that there's a number of predicted motifs that seem to be more common in our treatment effect genes than in the background genome as a whole, particularly ABREs, related G-box proteins. So these are genes we think are involved in ABA signaling in response to stress. And it's kind of neat that some of the ABRE genes, so ABRE genes are both enriched among upregulated genes and downregulated genes. I think suggesting the complex nature of some of these promoters, in which clearly context dependency is important, whether or not the effect of a presence of a promoter is going to lead to an upregulation or a downregulation. So transcriptome data, as I told the students on Monday, is great at identifying big, long lists of genes that show a treatment response. But it'd be nice if we can try to winnow down that list of genes to the genes we think really matter in driving functional differences. So the approach we took to sort of combine our expression data with our physiology data was to use principal component analysis. And the first step in this was to take our large list, our big list of genes. So here's 10 accessions, 20,000 genes we scored, and looking at the response of those genes. So this is the difference between treatment and drought, makes up this big matrix. We then use principal component analysis to decompose the variance in all of those 20,000 genes to a smaller number of variables. In the case of the spring analysis, we were able to turn 20,000 genes into just 5 variables. And those 5 variables explain more than 70% of the variation in the starting matrix. We then asked whether or not the principal components for gene expression showed correlations with specific physiological traits. So in this case, I'm showing you that there's a strong correlation between spring expression PC2, principal component 2, with this measure the phenotype we measured, specific leaf area. And it's important to remember in this case that positive and minus I don't think mean a whole lot in the context of these PCs. So a positive and a negative relationship, I can't quite clearly think what that means. Regardless, looking at these principal components with strong correlations with physiological traits, we asked whether or not those the genes that were most strongly loaded on those principal components were enriched for particular functional categories of genes using the MapMan ontology. So to highlight one of those cases, the specific leaf area correlation with principal component 2, we found that PC2 is strongly enriched for genes involved in photosynthesis, which might make some sense if you consider that in many plants, variation in specific leaf area is associated with relative growth rate. So we might have here is a short list of genes showing variation in natural populations of Arabidopsis that underlying variation in important phenotypes involved in growth rate. I want to touch on just a few general observations now looking at our genes that showed a genotype by environment interaction. There were much fewer functional enrichments in this group. We found enrichment for upregulation of the so-called cysteine-rich receptor-like kinases, which are also the domain of unknown function 26. We also found enrichment for histone synthesis and targeting to the chloroplast. Now, we didn't see any enrichment in the G by E genes for particular promoter motifs. And that may not be surprising if you think that in this analysis, at least, we were restricted to looking at motifs annotated in the Columbia accession. And it could be that there's a lot of variation between the accessions in the existence of these motifs. And so it'll be interesting as the resequencing data from these lines come online to go back and revisit and see if we can't try to get a better handle on the genetic control of these G by E genes at the level of cis. So to summarize my first part of the talk, I've shown you that there actually is a lot of variation in physiological responses of Arabidopsis to the soil water drying treatment we imposed. And so one important question to carry into the future is asking whether or not this variation is actually adaptive. Are there differences in fitness that underlie these differences or do these traits underlie differences in fitness? Second, I've shown you that expression variation can, at least in part, explain phenotypic plasticity. So one question moving forward, and I'll address this briefly in the next part of the talk, is how is expression variation in these genes controlled? Okay. So in the second part of my talk, I'm going to take the transcriptome data from the last part of the talk, combine it with association mapping approaches to ask three questions. First, sort of from a practical perspective, do our transcriptome experiments actually mean anything in a natural setting? So do the results from our growth chamber experiments actually represent what might be happening in the field? Second, I'll be able to ask what type of molecular variants underlie adaptation to climate. And finally, we'll just have a little peek at what role selection might be having at these loci that are associated with climate differences in Arabidopsis. So the approach we used was to take the transcriptome data from the first part of the talk. In particular, what we're carrying forward is the genes that showed a significant environment response. So these are treatment responsive genes. And those genes that showed genotype by environment interaction. And we did this for both early flowering and late flowering accessions, both together and in separate analyses. And for purposes for today, I'll just tell you about the combined analysis. The climate variables we're interested in were climate variables that we think might be important for describing the plant-water relations across the natural environment of Arabidopsis. So using climate databases, we came up with 5 climate variables. The first is variation in mean monthly precipitation, so just month to month. And then we developed models that predicted when the plants might actually be growing in their native habitat. And based on those predictions, we're able to calculate mean monthly precipitation during the growing season, the variation in this parameter. We estimated the mean vapor pressure deficit possibly experienced by the plants during their growing season. And we looked at the interannual variation in the mean monthly precipitation. So how much variation is there from this March to next March to the following March and so forth. And these are using climate data for about the last 50 or 60 years in the native range. Okay. The approach we used was to take the same 215,000 SNP data set that was used in the 2 earlier association mapping approaches and look at about 1,000 accessions of Arabidopsis. So starting with 215,000 SNPs, we asked if there are association between individual SNPs and the 5 climate variables I've just described to you. So this is represented here by our little red dots, which are the red bars. So these are SNPs associated with climate. We then looked for genes within 5 kb of a significant SNP and asked whether our genes showing a significant environment or G by E expression effect from the last part of for the first experiment were enriched in the tail of the distribution of those SNPs. Okay, so we're looking for genes that seem to be over-enriched in the SNPs showing significant climate association. So let me walk you through, it's really light, what these data look like. So I'm showing you fold enrichment of a particular class of genes, either up or down, over-enriched or under-enriched. And you're seeing 2 things. The light blue dots are those genes showing a significant environment effect in a transcriptome analysis. The purple dots are those showing a significant genotype by environment interaction effect. And so the first thing that jumps out at you is that genes, so that the SNPs with a significant climate association are enriched for genes that show G by E effects for a coefficient of monthly precipitation, coefficient of variation in precipitation during the growing season, and just marginally not so for a coefficient, interannual coefficient of precipitation. So what this suggests is that climate variables that describe variation, that is to say unpredictability in climate, are enriched for you tend to find genes that show genotype by environment interaction. I'm sorry, I'm not describing that very well. By contrast, genes showing a significant treatment effect are not enriched in any of these categories. So one interpretation of that might be that while treatment effect genes represent sort of a conserved treatment response across the distribution of Arabidopsis, the G by E genes reflect local adaptation to climate. Another possible implication of these results are that SNP variants that are closely linked to genes showing expression effect, let me restate that, that SNP variation, that SNPs showing significant climate associations are nearby to genes showing gene by environment interaction, suggesting that perhaps cis regulation is responsible for describing variation in those genes. To look at this data a slightly different way, asking if genes showing a treatment effect or a G by E effect showed higher than expected FST. So FST is a measure of sort of the structure of the localized structure of variation. We see that G by E genes do show a higher than expected amount of FST, suggesting that that variation shows some geographical structure. By contrast, taking a measure for the evidence of recent selective sweeps called pairwise haplotype sharing, we see that G by E genes, if anything, are under-enriched for this evidence for selective sweep, whereas treatment effect genes are ever so slightly enriched for evidence of recent directional selection. So to summarize the second part of the talk, I hope I've shown you that variation in expression may underlie local adaptation to climate. In particular, variation in cis regulation of expression may be underlying local adaptation to climate. Now, the Hancock paper I described at the outset found some evidence that there are evidence for coding sequence differences between explaining climate variation. So it'll be interesting to try to piece out in the future what the relative contributions of those two types of variants might be in climate adaptation. Second, and satisfyingly from my perspective, we were able to show that our lab-based assays of transcriptome function may be finding ecologically meaningful patterns in expression response. And so looking forward, we're interested in trying to increase the power of these kinds of tests. So actually this fall, I'll be doing a similar experiment in brachypodium with a much larger number of accessions. And we're hoping that by using basically more biological replication, we might be able to increase the power of detecting these meaningful expression differences. So in the final part of the talk, I want to focus in on a much more gene-based approach. In particular, asking which components of the drought stress signaling pathway vary in natural populations, and how natural selection is acting on those genes we think are important for the drought stress response. So I'm going to show this wildly oversimplified view of how of ABA signaling, just to illustrate a point. So this is certainly missing a number of sort of feedback interactions, but I think the basic flow of information here is correct, which is to say that we have a good understanding that plants somehow sense water deficit, that leads to abscisic acid synthesis and release. This ABA at the cellular level is recognized by the so-called PYR or PYL proteins, which inhibit a family of protein phosphatases, which in themselves, in the absence of an ABA signal, inhibit these kinases. And that some work has demonstrated that there's a direct interaction between these kinases and some transcription factors, and probably an indirect interaction for some between these kinases and some other transcription factors. But these transcription factors are the genes that are responsible for really affecting the stress response. So activating or deactivating genes involved in biosynthetic reactions, chaperonins, and so forth. So we were interested in asking how does natural selection act on these different classes of proteins? So looking at just a small subset of these genes, we looked at the role of selective, if there's evidence for selective constraint. And our metric in this case was looking at the ratio of non-synonymous to synonymous substitutions in the coding regions of these genes. So typically, values that are significantly below 1 are interpreted as indicative of evolutionary constraint or the action of purifying selection, whereas those values exceeding 1 indicate that there are more non-synonymous substitutions than you might expect, suggesting directional or positive selection is acting. So to look across the pathway, you can see that sort of the core proteins show a relatively strong signature of selective of constraint, which is say they have these DNDS values much less than 1. And this is also true for the small number of the sort of downstream effector genes, the chaperonins, the enzymes that I've looked at. An interesting contrast to this pattern is in this transcription factor family right here, the so-called AREBs. And for 2 of those transcription factors, we find a significant excess of non-synonymous substitutions. In fact, we can reject selective neutrality at those loci, suggesting that possibly positive selection has acted on those genes in the recent past. It's also interesting to note that the DREBs, which here I'm only showing 3 genes, but John McKay's group here has shown that a number of those transcription factors also show strong departures from neutrality, suggesting that positive selection may be acting on some of the DREBs as well. So while 2 of these AREBs show this highly elevated rate of non-synonymous substitutions, the third AREB, AREB1, shows a significant excess of polymorphism segregating its promoter. Okay, so this is just looking at how much diversity we see between accessions in the promoter region, while the other 2 are show about 10-fold lower diversity. So for the remainder of the talk, I'm going to focus on this first gene, AREB1, and try to understand what are the processes underlying that incredible excess of polymorphism. So what are AREBs? Well, we actually know quite a bit about this due to the work of the Shinozaki group in Japan, and as well as some others. And AREBs are transcription factors that recognize the so-called ABRE motifs, so the ABA binding response element. And we know in the Columbia genome that well over 2,500 genes have this predicted core motif of ABRE. Now, you recall from a minute ago, our transcriptome work found that the drought responsive genes in Arabidopsis were strongly enriched for these ABREs in their proximal promoters, suggesting that ABREs play a widespread role in transcriptional response to drought stress. So one hypothesis might be that variation in AREB expression of AREB itself could have large downstream effects as enacted through many ABREs scattered throughout the genome. So what does AREB itself look like? So here's just a little simple gene model of AREB1. AREB1 has an exon right here that's pure untranslated region. And the start codon is actually right here at the beginning of exon 2. So we started sequencing a bunch of Arabidopsis accessions. The first thing that jumped out at us was that there's a large number of small insertions in this intron 1, which we're sort of thinking is probably being part of the promoter sequence itself. In particular, there's a 5 amino acid insertion right here at the very beginning of the coding region of AREB1, as well as these 3 other small insertion events relative to the Columbia sequence, and then a large 500 base pair deletion relative to the Columbia sequence. Now, there's probably variation in lots of genes in the Arabidopsis genome. But what's interesting about this case is that if you look at a given accession, and we've looked at a whole lot of them now, accession either has or lacks all 4 of these small insertions. Okay? So these are so-called plus alleles and minus alleles moving forward. And what's even more interesting is if you look across Arabidopsis, so here's 200 and some accessions, that the plus alleles, those having these extra pieces of DNA, the minus alleles, those lacking, are segregating at nearly equal frequency in the Arabidopsis populations. This is a very unusual pattern. But natural selection, so partially because I think most people think of natural selection as playing a role in removing variation from populations. That is to say, some new variant shows up, is deleterious for function, it's swept out of the population, or some rare variant appears that's beneficial for function, it's swept to fixation of the population. So generally, natural selection plays to reduce variation in population. But there are some interesting cases where that's not the case. And one of the best studied examples in Arabidopsis is the role of in the pathogen response genes. So this is sort of a classic case where the basic idea is that in the presence of some pathogen, having this resistance gene, of course, confers a benefit. But in the absence of pathogen, expressing that gene leads to a fitness penalty. So you end up with this balanced situation where you either do or you don't want to have this gene. And that's classically referred to as balancing polymorphism, balancing selection leading to a balanced polymorphism. And that process leaves a characteristic signature on the sequence of the gene in question. So the classic pattern is to see these 2 deeply diverged allele classes with very little variation within each allele class. You also typically see elevated polymorphism right on top of the inferred balanced polymorphism. So in this case, they're invoking that there's a polymorphism right here that's being maintained by selection, And that nucleotide diversity is enriched right there because you have these two deeply diverged classes of alleles. And as you move away from that polymorphism, the recombination basically has more chance to sort of break up the associations, and the polymorphism decreases. And a similar pattern is that you typically find high linkage disequilibrium, which is to say that a SNP here is highly predictive of the SNP state on the other side. So high LD, high SNP diversity, deeply diverged allele classes. So while balancing selection has been frequently observed in the context of biotic interactions, it's less clear what that process might have to do with genes that we think are nominally involved in abiotic stress response. So to address this in AREB, we first looked at the sequences. So do we see this pattern of a balanced polymorphism in the AREB1 sequence? So again, here's the structure of the gene, upstream gene, AREB1, start codon here, reading that direction. And again, we see the typical case where extremely high polymorphism right on top of the site of interest. And I'm showing you here the background rate of polymorphism across the genome. And that sort of tails off as you move away this way. Tails off very quickly as you move this way, possibly because there actually is still some purifying selection acting on the actual coding sequence of AREB1. Whereas in the promoter region, some variants are allowed to hang around at higher frequency. Looking at the genealogical relationships among accessions, looking at this sequence right here, we again see this classic pattern. Two deeply diverged allele classes. And this corresponds very perfectly with the presence of these insertions in intron 1 and the absence of those insertions in intron 1. We also see highly elevated linkage disequilibrium. So looking here at the AREB1 intron and then moving about 70 kb downstream of AREB1 towards the center of the chromosome, 30 kb upstream of AREB1, we see that actually this pattern of LD extends a little bit downstream of AREB1. But there's a really strong signal of linkage disequilibrium right over the intron and AREB1. So to summarize the sequence component of this, we do seem to see evidence for a balanced polymorphism in AREB1. Deeply diverged allele classes, elevated linkage disequilibrium, these two frequency classes segregating at two allele classes segregating at high frequency, and the high nucleotide polymorphism. So how does all this natural variation, this possible balanced polymorphism, actually affect AREB expression? Well, we can look at the sequence of intron 1 to try to develop some hypotheses about what those differences might entail at the functional level. So I'm showing you two alleles. Here's one representative of the so-called plus class alleles, and those one representative of the minus class alleles. And you can see the four insertions I mentioned earlier. So here's one spanning this line, there's two, there's three, and here's the fourth one in the coding region of AREB1. Now, I've highlighted predicted promoter binding motifs. So it's important to emphasize none of these have been experimentally validated. But these are just sequence motifs that we think might be associated with the action of particular transcription factor binding events. In black, I'm boxing in motifs, predicted motifs that are conserved between the two allele classes. And in color, I'm showing cases where the motifs differ. And that's usually due to the presence of a polymorphism between the two allele classes. In particular, we see these three MIB transcription factor binding sites, which are present in the Columbia type, the minus alleles, but absent in the plus alleles. Now, these MIB transcription factor binding sites are of interest because some evidence suggests that the transcription factor that's predicted to bind to those sites is involved in drought stress ABA-mediated signaling. Now, while the plus class lacks those three sites, instead it has two motifs that are predicted to be binding sites for this gene called A-gamma-SLIC15. Now, A-gamma-SLIC15, it turns out, is also ABA responsive, but it's generally understood to play a role in seed development and is highly expressed in the siliques. So that suggests that the AREB1 variation we see, expression therein, might be represent this classic balance between two roles of ABA. On the one hand, the drought stress response. On the other hand, a developmental role for ABA. So to test this directly, we're using a transgenic approach. I've taken the two different kinds of alleles, I've attached them to a reporter gene, and then we've knocked those constructs into an Arabidopsis Columbia accession wild type plant. And we're in the process of trying to score the timing and the expression both constitutively and in response to stress. And those experiments are probably to come this fall, but I do have a little bit of preliminary data I'd like to share with you. So looking here, if insertions are present, the Columbia type, you can see that the promoter for AREB1 is driving expression throughout the cilique. Whereas in the absence of those, when the insertions are present, you see that expression of GUS is restricted just to the distal part of the growing fruit. And I'm showing you two different controls here at the bottom, constituent overexpression and then, of course, empty vector. Now, I want to say these are preliminary data. This is a very small sample size. But I think this is potentially an interesting phenotype to be looking at because one of the strongest responses we see in Arabidopsis to drought stress is a shortening of the fruits. So there's a strong impact of drought stress at the level of the cilique. And we're possibly uncovering some of the molecular variation that may describe variation in that response in Arabidopsis. So to take a broader look at how this variation might be affecting the role of AREB in response, I've cloned the entire fragment, so from the upstream gene all the way through AREB1 coding sequence of the different allele classes and transformed those into an AREB1 tDNA knockout. And right now, back at UT, we're growing an experiment of these different alleles and some of the classic genotypes to look at whether or not variation in AREB1 is leading to differences in growth, in physiology, and then in the whole transcriptome response to stress. And we're going to look at that the transcriptomes using RNA sequencing. So to conclude this third section of the talk, I hope I've shown you that the AREB transcription factors show this classic pattern of diverging selection pressures. And we're interpreting this as a sign of a balanced polymorphism acting in intron 1 of AREB1. And finally, I've shown you some preliminary evidence suggesting that promoter variation in AREB1 might lead to distinct expression domains for that gene. So to summarize overall, the first part of the talk, I showed evidence that variation in expression may underlie variation in phenotypic plasticity in response to drought stress in Arabidopsis. The second part of the talk, I showed evidence that expression differences in Arabidopsis may be involved in local adaptation to climate. And this may be due, at least in part, due to variants acting in cis. And finally, I'll suggest that AREB1 might be the first known example of balancing selection maintaining diversity in an abiotic stress response gene. So all the work I've told you about today is part of a multi-year collaboration between myself, Tom Younger, John McKay, and Jim Richards, partially funded by the Arabidopsis 2010 project. The genome association mapping work was done largely by a now former PhD student at UT Austin, Jesse Lasky, and also in collaboration with Tim Kitt. And I need to acknowledge the great work over many years of a number of undergraduates for their help in the lab. And I'm currently funded by the USDA. And I'd like to take any questions you have.